Hi, today we're going to take a look at transport across the membrane. Specifically, we're going to be taking a look at passive transport. So let's get started. Recall that the plasma membrane is a semi-permeable membrane. That means that some things can freely pass through the membrane while other things cannot. The substances that can freely pass through the membrane are small molecules such as oxygen or carbon dioxide, even water. And things that can't pass through the membrane are things like ions, anything that is hydrophilic, and of course large molecules are definitely not going to be able to pass through the plasma membrane. Let's take a look at the three types of passive transport. There are three types of passive transport. There is diffusion, osmosis, and facilitated diffusion. Now diffusion, when we're talking about diffusion, we're looking at solutes or particles. With osmosis, we're specifically looking at water. And in facilitated diffusion, we're looking at slightly larger molecules moving through a transport protein. And we're going to take a look at each of these three right now. Diffusion is the movement of solutes or particles from an area of high concentration from an area of low concentration. It requires no energy. So all of the forms of passive transport will not require any energy. So diffusion we see here, we have in green are solutes, and they are moving from an area of high concentration down their concentration gradient to an area of low concentration. That's going to happen with, whether it be a gas or whether it be a dye. So the molecules are going to move, or the solutes are going to move from their area of high concentration to their area of low concentration. We are going to take a look at a short video on diffusion. Roll this up so we can watch it, and here we go.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
So that video just showed a short clip on how diffusion works. We saw that the sugar molecules move from an area of high concentration to an area of low concentration, and they diffuse throughout the beaker until we got a solution where we had an even distribution of sugar molecules throughout the beaker. Let's take a look at another specific type of diffusion, and that is osmosis. Osmosis is the diffusion of water across a membrane. That's important. It specifically is dealing with water, and it's dealing with the movement of water across a membrane. Again, water is moving from an area of high concentration to an area of low concentration or down the concentration gradient. Here we've got a diagram that's showing that we have a lot of water on the left-hand side, and water is moving down the concentration gradient to the right-hand side. So we've got water molecules in blue moving across the semi-permeable membrane from an area of high concentration to an area of low concentration. We're going to take a look at another animation dealing with osmosis as well. Here we go, and play.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So those vocab words you will need to know, hypertonic, hypotonic, and isotonic. So those are examples of cells in different types of solution, and we're going to take a look at what happens to a cell when you put it in either an isotonic, a hypertonic, or a hypotonic solution. So here we have a cell in an isotonic solution, and what we notice is that the water concentration in the environment right here, okay, is equal to the water concentration inside, the water concentration in the environment is equal to the water concentration inside the cell. So what we're noticing as we do this is that water is going to be moving both into and out of the cell at equal rates, and the cell is going to stay happy. It's going to be in a stable environment. So the cell is what we call at equilibrium, because the water in the environment and the water in the cell are equal to each other. Even though water is moving constantly into and out of the cell is happy. So it's going to stay, stable, if you will. Let's take a look at what happens to a cell in a hypertonic environment. So what we're going to notice here is the water in the environment, right here, is less than the water in the cell. So what's going to happen is water is going to move from an area of high concentration inside the cell to an area of low concentration outside the cell or in the environment. And what's going to happen to the cell is going to shrivel up. Cell is going to shrivel up. This term is called plasmolysis. So the cell is going to shrivel up. Let's take a look at a cell in a hypotonic environment. So what we're noticing in a hypotonic environment is that there is more water here. There is more water here in the environment than there is in the cell, and water is going to move from an area of high concentration to an area of low concentration. So water is going to move into the cell, and when water moves into the cell is going to swell. And when the cell swells, it's possible that it might undergo something called cytolysis. And that word means Cyto means cell and lysis means to break, so that literally means cell breaking. So if enough water goes into the cell and the cell swells and it's not able to regulate how much water is actually moving into the cell, it's possible that the cell will burst. So let's take a closer look at what happens to animal cells or plant cells in these different types of solutions. So let's take a look at an isotonic solution here first. So our isotonic solutions, here we have an animal cell, it's a red blood cell. We see that in an isotonic solution where there is equal amounts of water out of the cell and in the cell, water is moving at a constant rate into and out of the cell, and the cell appears normal and happy. Same thing with a plant cell. If a plant cell is placed in an isotonic solution, water is able to pass through both the cell wall and the plasma membrane. It's passing through at equal rates, and the cell is what we call flaccid. If we place a cell in a hypotonic solution, that means that there is more water in the environment than there is in the cell. Water is going to move from an area of high concentration in the environment to an area of low concentration in the cell, and water is going to rush in, and if the cell can't regulate that, such as animal cells, they can't regulate that, it's possible that the cell will actually swell up and burst. So here we see the cell lysing or breaking. With a plant cell, we won't see the plant cell burst, but the plant cell will become what we call turgid. And that means that the plant cell is going to swell up, but because we have that nice cell wall, the plant cell will not actually burst. Let's take a look at a cell in a hypertonic solution. So in a hypertonic solution, that means that there's more water in the cell than there is in the environment. So water is going to move again from an area of high concentration to an area of low concentration outside the cell, and the cell is going to shrivel up. With a plant cell, we see that again, water is going to move across the membrane and across the cell wall out into the environment, because there's more water inside the cell than in the environment in a hypertonic solution. But again, what's going to happen here is that plasma membrane or the cell is going to actually shrivel, but the cell wall is going to stay in place because the cell wall provides structure and rigidity for plants. So the plant cell wall is actually going to stay in place, kind of create a little skeleton, but the actual cell membrane is going to pull itself away from the cell wall and it's going to shrivel up inside. When a plant is wilting, if you will, falling over, if you ever see plants wilt, this is what's happening inside the cell. If a cell gets a lot of water and starts to stand really tall, we call that turgid. And when a plant is kind of in that in-between stage, sort of, if you will, coming back to life, if you've ever forgot to water a plant and then slowly you see after you've watered it that it's slowly kind of starting to come back to life, getting to that turgid phase, that's what we call flaccid. So again, you need to know what happens to either an animal cell or a plant cell in these different types of solutions. So isotonic, hypotonic, or hypertonic. Facilitated diffusion is another type of passive transport, and it's a type of passive transport that uses transport proteins to move materials from a high concentration to a low concentration. So there are some cases where there are molecules, so for example glucose and amino acid, that are slightly too big. So remember that these are monomers. They're slightly too big to pass through the membrane, the lipid bilayer, here our phospholipid bilayer on their own, but they need help, they need assistance, and that's what that word facilitated means. Is facilitated means to help. So they're going to use a transport protein to help them move across the membrane from an area of high concentration, as we see here. So we're moving again from an area of high concentration down our concentration gradient to an area of low concentration. And what's unique about facilitated diffusion is, again, the use of that transport protein. Take a moment to answer the following questions, whether or not these statements are true or false. So statement number 1 is, in diffusion, substances always move down a concentration gradient. Statement number 2, osmosis is a form of diffusion. And statement number 3, the process by which molecules diffuse across a membrane through a protein channel is called active transport. So take a moment right now to identify whether or not each statement is true or false. All right, statement number 1 is true. Statement number 2 is also true. And statement number 3 is false. The process by which molecules diffuse across a membrane through a protein channel is not called active transport, but is called facilitated diffusion. Very good. Moving on. Just to recap, there are 3 types of passive transport. There is diffusion, osmosis, and facilitated diffusion. In diffusion, you are looking at solutes, so particles or molecules moving from an area of high concentration to an area of low concentration or down their concentration gradient. Osmosis, you are specifically looking at water moving across a membrane. Again, water is going to move from an area of high concentration to an area of low concentration or down its concentration gradient. There are 3 key words that are associated with osmosis, and those are isotonic, hypertonic, and hypotonic. In all types of passive transport, no energy is required by the cell. So the cell is able to get substances into or out of the cell without having to expend any energy. And again, with all types of passive transport, substances are moving from an area of high concentration to an area of low concentration or down their concentration gradient. That concludes our presentation. Good luck.